you understand the human brain? Do you know what the structure inside the brain looks like? Every single thought and emotional fluctuation you have is intricately linked to the brain. The brain, as the most advanced organ of humans and the most alluring one, is so complex, and its rational control makes it like a supreme commander of reason, ensuring the orderly operation of all human functions. Therefore, the brain is of vital importance to humans. To unlock its mysteries, generations of scientists have been tirelessly researching and exploring. Later, we gained a certain understanding of the brain.
Do you want to know the history of brain science? Research now, I will explain it to you in detail.
Back then, scientists had long established a rule. Whether it's a towering tree, a tiny cat, human skin, or the heart, all are built from individual cells. Just like a house is built from bricks. The brain, however, was an exception. When brain tissue was sliced thin, stained, and placed under a microscope, researchers didn't see distinct cells. Instead, they saw a tangled mess of fibers, like an overgrown bush with intertwined branches, making it impossible to distinguish individual units. Everyone was perplexed. Could the brain really be an exception to the cell theory? An Italian doctor named Golgi crashed through the first door to solving this puzzle. At a makeshift lab on his kitchen counter, fresh out of medical school, and working at a small-town hospital, he had no research funding or proper equipment. He bought a microscope with his own money and set up a simple lab in his kitchen. One day, he immersed brain tissue hardened with potassium dichromate into a silver nitrate solution. Suddenly, clusters of black little corals emerged on the previously muddled slices, rarely stained nerve cells, with their delicate branches clearly visible. This became the famous Golgi staining method. Yet Golgi misinterpreted what he saw. Staring through the microscope, he thought, thoughts travel as fast as electricity through wires. If there were gaps between cells, how could signals transmit instantly? He was convinced that invisible threads hidden in the unstained areas connected all nerve cells into a large, continuous network. This was the reticular theory of the time. Unfortunately, the staining method was highly unstable and gained little recognition. Though Golgi later discovered the Golgi apparatus, he left the field of neuroscience with regrets. This crucial key lay in use for 15 years. Cajal, the brain's artist and truth seeker, it wasn't until a young Spanish man picked up the torch. Santiago Ramon y Cajal. As a child, Cajal was a rebellious troublemaker for his father, preferring to squat on the ground observing insects and drawing flowers rather than studying textbooks. His father dismissed art as unpromising and forced him to study medicine. No one could have guessed that his artistic talent would later become an invaluable asset in brain research. When Cajal first started working, his salary was meager, so he bought a cheap microscope and set up a lab in a corner of his home. In 1888, he heard about the Golgi staining method, immediately gathered materials to experiment, and secretly improved it. He used double impregnation to stabilize the staining and focused on the brains of young animals. Their neuronal structures were clearer and easier to distinguish than those of adults. In the years that followed, Cajal acted as a brain artist, devoting his artistic talent to unlocking the brain's mysteries. He often spent over 10 hours a day, hunched over his microscope, sketching every detail he observed. Over his lifetime, he created nearly 3,000 hand-drawn brain illustrations, capturing even the tiniest protrusions on neural branches. It was in these sketches that he made a cognition-altering discovery. There was a small gap between neurons. The tail of one neuron, later named the axon, never fused with the branches of another, later called dendrites. No matter how close they got, Cajal suddenly realized the brain was not a continuous network, but composed of billions of independent individuals, neurons. They transmitted signals through close contact, not by being welded together. This was the neuron doctrine. Few believed him until 1906, when the Nobel Prize in Physiology or Medicine was awarded jointly to Golgi and Cajal. The award ceremony took a dramatic turn. Golgi, holding his trophy, began his speech by directly refuting Cajal, claiming the neuron doctrine was wrong and the brain was indeed a continuous network. Cajal refused to back down, responding in his speech the next day. Nature never simplifies its complexity for the convenience of human understanding. This academic debate on the Nobel Prize stage became a classic moment in scientific history, unveiling the truth. Synapses and signal transmission. The truth was finally confirmed 50 years later with the invention of the electron microscope, which could visualize structures as small as 0.1 nanometers. Scientists clearly saw a 20-nanometer gap between neurons. They named it a synapse. Cajal was right, but new questions arose. If neurons weren't connected, how did signals jump across synapses? scientists split into two camps. The spark school argued, signals are like electric sparks, leaping across synapses directly. Otherwise, how could they transmit so fast? The soup school disagreed. If it were only electricity, why would stimulating different nerves produce opposite reactions in the same organ? There must be chemical messengers traveling across synapses. On the eve of Easter 1921, pharmacologist Otto Loewy had a vivid dream. In it, he conceived an experiment. First, he electrically stimulated the vagus nerve of a frog's heart, which slowed its beating. He then collected the fluid around the heart and dropped it onto another unstimulated frog's heart. And the second heart also slowed down. Loewy woke up in the middle of the night and scribbled the idea down, but the handwriting was illegible the next morning. He was devastated, until the same dream returned the following night. This time, he rushed to the lab in his pajamas and performed the experiment exactly as he dreamed. It worked. The chemical messengers in the fluid were later called neurotransmitters. The spark school wasn't entirely wrong though. In the 1950s, they discovered electrical synapses, gaps only 3 nanometers wide that allowed rapid electrical signal transmission. These synapses are rare, however. Most signal transmission relies on neurotransmitters, the soup. As for how signals travel inside neurons, in 1939, Alan Hodgkin and Andrew Huxley found an unlikely helper, the giant squid. It has a massive axon nearly 1 millimeter in diameter, thick enough to insert microelectrodes directly. Through studying this axon, they discovered a key principle. Electrical signals and neurons follow an all-or-nothing rule. Either a signal is sent at full strength, or it's not sent at all. No halfway states. Like binary code, complex information is transmitted through combinations of different firing frequencies and patterns. Even more fascinating, psychologist Donald Hebb proposed that neurons that fire together wire together. It's like a path that becomes wider and smoother the more people walk on it. When you memorize a poem or learn to ride a bike, neurons in your brain strengthen their synaptic connections through repeated activation. This idea unexpectedly became a source of inspiration for artificial intelligence. The underlying design logic of modern deep learning models, capable of image recognition and language translation, stems from Hebb's rule. By understanding our own brains, we've created silicon-based bionic brains. The endless journey of exploration, looking back over more than a century of discovery. From Golgi's accidental breakthrough in his kitchen to Cahal's nearly 3,000 brain sketches. From the heated debate on the Nobel Prize stage to the clear visualization of synapses under electron microscopes, humans have been using their brains to unravel the brain's mysteries. This 1.4 kilogram organ holds hundreds of billions of neurons, equal to the stars in the Milky Way. Every synaptic change, every signal transmission, holds the secret of what makes you you. The exploration of the brain has no end. To this day, we continue to peel back the layers of mystery surrounding the most complex organ in the universe.